guys, Dr. Biology here, and this is Revised with Dr. B. It's week 5 and I'm going to go through bioenergetics. So these are the topics I'm going to go through. It's quite a small unit, but a really important unit in terms of your understanding of GCSE biology. So how it works, I'm going to go through the main points, not everything, just some of the important bits. I'll give you signposts to further resources, particularly detailed videos, past paper exam questions, and also I'm doing weekly videos up until your exams, so please do subscribe. Okay, first of all, photosynthesis, you will need to know the equations. If you're doing foundation tier, you just need to know the word equation. If you're doing higher tier, you need to know both the word and symbol equation. One of the key things they do like is you knowing what the symbol equation for glucose is. C6H12O6. Photosynthesis, so obviously you need water, you need light as an energy source, and you need carbon dioxide. So water's coming from the roots up the stems into the leaves. Carbon dioxide enters through the stomata of the leaf, and obviously chlorophyll traps the light in the chloroplasts, and then it makes, that's energy to make sugars, make food. It releases oxygen. Now, some of that oxygen is released into the air, but some of it could be used for respiration. And obviously sugars are made and then they can be converted into starch. So the stored food in other parts of the plant. And you should know that starch turns iodine dark blue or black. Okay, so you might have done this experiment in your school. So this is testing leaf for a starch. This is not a required practical, but they do like you to know this experiment. So here you can see leaves and you can see bits of cardboard being put over the different leaves to stop light getting to it. So you will know if you've done this, you need to know the method generally. So you boil a leaf for about a minute. You then turn the Bunsen off. You put the leaf into back in the boiling tube, into a boiling tube with ethanol. You turn off the Bunsen burner because ethanol is flammable. Okay, and then you should know plants contain a green substance called chlorophyll, absorbs sunlight so plants can make their own food. So and it's stored as starch. So testing a leaf for starch, then you remove it. You can then put it back in the hot water to soften it up, and then you spread it out, cover it with iodine, and if the iodine goes blue-black, there is starch in the leaf. That tells us that the plant has been photosynthesizing. Here's a few parts of the experiment and purpose of the part of the experiment. You could pause this now and have a go yourself. Okay, so here's the answers. There we go. Right, the required practical, you definitely need to know the required practical, and what I suggest is that you watch my video, okay, and also do some questions related to it. So the video goes through the main bits of the method and talks about what the different variables are and some features of the experiment. If you're doing higher tier, you will need to know the inverse square law. They sometimes show you the inverse square law in the exam, but as you can see, it's 1 divided by distance squared. Okay, so you need to always convert the centimeters into meters. Okay, sometimes they'll give you the distance in centimeters, convert it into meters, and then you can work out how many units, arbitrary units there are. But the main thing you need to remember in everything you do related to inverse square law is this. Light intensity obeys the inverse square law. This means that if you double the distance, you quarter the intensity of light. You'll need to know about limiting factors. There are 3 that they want you to know: light, carbon dioxide, and temperature. Light and carbon dioxide are very, very similar. So as you can see with light, as light increases, so does the rate of photosynthesis, although eventually it reaches a plateau, or it levels out, or it reaches a point where it does not increase any further. Note it's not stopped, it's still producing sugars through photosynthesis, okay, but some other factor is limiting at this point, at the plateau. So it could be carbon dioxide, it could be temperature. Exactly the same with carbon dioxide. Temperature's a bit different because it's related to enzymes. So enzymes are really important in photosynthesis. So you can imagine with an increase in temperature, you reach an optimum. Then any further past the optimum, then enzymes will become denatured. What's glucose used for in plants? Well, they're used in respiration to release energy, joined together to make starch for storage. They're combined with nitrates from the soil and they make proteins that allows new growth. Joined together to make cellulose for cell walls, so it strengthens the cell. Converted into fats and oils for the seed. And it can be converted to fructose for the fruit. So this means that things like animals or herbivores eat it and they spread the seeds. Really good way to remember this is crops. Cellulose, that's the cell walls. Respiration, oils and fats, proteins and starch, crops. Okay, on to aerobic respiration. So again, foundation tier, you only need to know the word equation, whereas if you're doing higher, you need to know both. Interesting, aerobic respiration is the opposite to photosynthesis, as you probably have noticed, and it's basically releasing energy, and it's chemical energy. Please do not use the word produce or make energy. It does not do that. Okay, it releases energy, provides energy. So they need energy for different things: growth, repair, mitosis to make new cells, movement, production of proteins, and active transport, to name a few. You will need to know about anaerobic respiration, so that's respiration without oxygen. So glucose without oxygen, it doesn't break down efficiently and it produces lactic acid and a little bit of energy. Okay, so when you're exercising, all right, you need to know about responses to exercise. So your heart rate, your breathing rate increase, your depth of breathing increases. What you're trying to do is increase the blood flow to your muscles. So it's delivering glucose and oxygen to your cells for respiration, and then removing that carbon dioxide, and you're breathing out that carbon dioxide. Skin may go red as energy is released as heat energy, so more blood travels close to the skin surface to radiate excess heat. Muscles may feel painful, and we'll talk about that in a minute. So the incomplete breakdown of glucose produces lactic acid, so this builds up in the muscle cells and it stops the muscles from contracting efficiently. This causes fatigue, pain and cramping. So what you need to do is you need to repay what we call an oxygen debt. So to break down that lactic acid, you have to breathe deeply and get lots of oxygen into your body. Anaerobic respiration in plants is slightly different. So it produces ethanol, which is a form of alcohol, carbon dioxide and a small amount of energy. This can be used by humans effectively for making bread. So dough contains flour, sugar, yeast and water. Anaerobic respiration occurs and the dough will rise because of the bubbles of carbon dioxide produced. Also, we can use the ethanol, so that's to make alcohol. So you put yeast with a source of sugar in anaerobic conditions. You prevent oxygen getting into the bottle, and it ferments and it produces ethanol. Also, we can test for carbon dioxide as well, so not necessarily anaerobic respiration. So you can actually anaerobic respiration. You can see there's liquid paraffin, that's stopping any oxygen getting into the yeast and sugar. So what is produced is carbon dioxide. We can use an indicator solution, for example lime water, that will go cloudy when carbon dioxide is present. Lastly, metabolism. It's the sum of all reactions in a cell or the body of an organism, and reactions are fueled by energy through respiration. So you do need to know in humans, examples include reactions in respiration, building lipids or fats, and enzymes breaking down food and building new molecules. In plants, again, respiration, photosynthesis, and converting glucose to starch for storage. Obviously, lactic acid has to be removed, as I said, after vigorous exercise in anaerobic conditions. So it's broken down into carbon dioxide and water. In the liver, lactic acid is broken down into glucose, which is then stored as glycogen. And this is definitely higher tier in terms of the role of the liver. So the liver does several things like detoxifying poisonous substances, passing the breakdown products into the blood so they can be excreted into urine, that's urea, or breaking down old worn out blood cells, and removing lactic acid. So basically the lactic acid to the liver is converted back into glucose. So it's repaid once the oxygen debt is repaid once it's converted back to glucose, and then it's been completely broken down in aerobic respiration. And it can also be converted to glycogen and stored in the liver until it's needed. Exam Q&A packs, they can be found on my website on drbiology.uk. And I hope you found that really useful. Please do look at the required practical. I'll put the link in the text box below. Good luck. And week 6, I'm going to go into paper 2. I'll see you soon.